Welcome, and yes, we can start for the sake of the time. The talk of today is about genetics in inherited cardiovascular diseases, practical utility, and we are going to talk, giving especially some practical examples of how to deal and how we are practically doing genetics in these conditions. My name is Lorenzo Monserrat. I'm a bit older now than in this photograph, but I think you can recognize me. And I've been involved in the area of cardiomyopathies for many years, for example, in the classification of the European Society of Cardiology of the cardiomyopathies, in the genetic counseling and testing, in all these papers where we put together information, and also in clinical risk stratification. And I have worked for many years in hypertrophic cardiomyopathy, and in this paper, that is the new risk prediction model for sudden death in hypertrophic cardiomyopathy, based in clinical features. Our center in La Coruña, in the northwest of Spain, where I live, is the second center providing more patients after the center led by Professor McKenna and Perry Elliott. So with all this experience, and a number of years ago, 15 years ago, we started thinking that genetics could have a role in cardiomyopathies, channelopathies, and other inherited cardiovascular conditions. The problem for genetics is that it's not so easy. Complexity of the information and the volume of knowledge every day increasing make necessary multidisciplinary specialized teams we deal with this. We are not able to keep updated, and for me it was very clear that I was not able to read all the papers and have all the information, so we need knowledge management. And there is a continuous technological evolution that makes an additional challenge for everybody that's involved in this. So when we want to solve the problem, you cannot do it for yourself. And in that way, we consider at the time that we need to do commercialization and partnership and a global approach. Today we talk about these conditions: inherited cardiovascular diseases, including cardiomyopathies, hypertrophic, restrictive, dilated, arrhythmogenic cardiomyopathies, non-compaction, channelopathies, low QT, Brugada, catecholaminergic polymorphic ventricular tachycardia, idiopathic conduction system defects, Marfan and other vascular syndromes, and also we can talk about familial dyslipidemias. All these conditions have things in common. These common features are that they are very heterogeneous in the clinical presentation, all of them. Early diagnosis is difficult, and they are associated with sudden death risk, being the most frequent causes of sudden death in young individuals, but also in individuals not so young. And they are familial, so there is a role and a potential for genetic diagnosis. I will dedicate more time today, because of the lack of extended time, to talk about one example, which is hypertrophic cardiomyopathy, but if you want, in another moment we can talk about any other of the conditions. All of them have things in common. Hypertrophic cardiomyopathy is one of the most common causes of sudden death in young individuals, and we know it's a very heterogeneous condition, morphologically, clinically, and with respect to prognosis. And this heterogeneity of the disease clinically is related to heterogeneity in the genetics. Now we've got, for example, more than 1,000 mutations in sarcomeric genes, and this machinery of the contraction, the sarcomere, with many different pieces, is involved not only in hypertrophic cardiomyopathy, but also in dilated cardiomyopathy, restrictive cardiomyopathy, left ventricular non-compaction, even in congenital abnormalities like septal defects. So it is important to really know that we can find the mutations. And if you study, like previously with Sanger sequencing, the main sarcomeric genes, we will find mutations in approximately 60% of the patients with hypertrophic cardiomyopathy. But there are other causes. For example, mutations in cytidase protein genes that are involved also in dilated cardiomyopathy are responsible maybe between 2 to 5% of the cases of hypertrophic cardiomyopathy. But mutations in RAS-MAP kinase genes also cause this condition. Noonan, Costello, cardiovascular syndromes are responsible for approximately 1 to 2% of the cases of hypertrophic cardiomyopathy. And we have other different causes. Storage diseases like Danon, Pompe, Fabry disease, mitochondrial diseases, calcium regulatory proteins like phospholamban, and others. Is it really useful to perform genetic testing? Can we get any advance? And I will show you examples of this. This is a woman, 79 years old, hypertensive, 220 millimeters of mercury was her systolic blood pressure. The question here always for the clinician is whether we do a familial study or even if we do a genetic testing. This is a very old slide. This woman has a symmetrical septal hypertrophy, systolic anterior motion of the mitral valve. She had got high obstruction, subaortic obstruction, and also some apical hypertrophy that you see here with the ace of the spades even profile here in the apex. So for me, this woman, hypertension is an exclusion diagnosis for hypertrophic cardiomyopathy, but in this case it could be a combination of both of them. The family didn't come for the screening. This woman didn't undergo a genetic testing at that time. What happened is that sometime later I received this girl. This 18-year-old girl returned to be the granddaughter of the sister of the previous woman. And you see she's got a very severe asymmetrical septal hypertrophy, also affecting the anterior wall, and also has a mid-ventricular apical obstruction with a paradoxal flow toward the apex. And the question is, this woman, this girl, is related to the previous woman. The mother of this girl, which is in the middle, was normal. This is a very old slide. But the mother of this woman, one day I was doing echo because my role in the cardiology department was also in the cardiovascular imaging, and I received this woman because of a stroke with atrial fibrillation, and this woman is the grandmother of this girl with severe hypertrophy. And you see, she's got also asymmetrical septal hypertrophy, mid-ventricular obstruction, and paradoxal flow to the apex. So you will see the summary. We have two elderly women with hypertrophic cardiomyopathy, and we have a girl with severe disease. And in the middle, the mother was clinically normal, clinically and morphologically normal. In this case, with genetic testing by Sanger sequencing, the old and classical way of finding mutations, we found a mutation in troponin C that we consider likely or almost definitely pathogenic, and the mutation is present in all of them. The question now is this mutation really pathogenic when the mother is normal? The second question is, this mutation could explain the phenotype of these people, but this really explains the variability in the phenotype of the family? And the answer is that there is something else. And with next generation sequencing, we were able to test all the other genes that are related to the condition and to find a second mutation that is only present in the girl. This PTPN11 mutation has been previously described, and it's a mutation de novo. It was not present neither in the mother nor in the father, only in this woman. And this mutation has been described previously in 3 references, 6 unrelated carriers with Noonan and Leopard syndromes, always as de novo mutation like in this girl, not present in the parents, associated with severe hypertrophy. Café au lait spots were the only extracardiac feature that was clearly apparent in this girl. And even one of the patients described in the literature had an additional sarcomeric mutations. So in this family, you see that the genetic testing is able to explain the variability. The PTPN11 mutation is responsible for the severe disease in the girl, but not for all the disease. The apical paradoxal flow we have seen, which is present in the grandmother, is likely related to the troponin C mutation. And we think that this woman may develop late onset disease, but they don't have the same disease as the girl. As you can imagine, if we consider that family history of sudden death is a risk marker for the patients, imagine that you are seeing the sister or the brother and they have hypertrophic cardiomyopathy. It's completely different if they have only one or they have the 2 mutations. How we do it? Next generation sequencing, I will give you a brief overview of this technology. What we do is to take the samples, either blood, saliva, or DNA. When we get the DNA, then we cut the DNA, we make the fragmentation, either with ultrasounds, with the Covaris machine, for example, or with enzymes. Ultrasounds are more random and more interesting for us. And then we need fragments of a given size. If they are too big, they will not work well. If they are too small, we will not be able to put them in the puzzle we will have to put together in the bioinformatics. When we have the fragments, we usually do target enrichment. This is a process to select those fragments that we are interested on, and then we go to the sequencing, and when we have sequencing parallel millions of fragments, we are able to put them versus the reference sequencing with our informatics tools. Here you see the start of the process. We have the fragments, either DNA or RNA, and we hybridize with RNA libraries that have been prepared to hybridize with the fragments of interest, and they are coated biotin with biotin. Then we put them together, these hybridized fragments, with S-protein coated magnetic beads that will bind them, and then we are able to capture the fragments of interest with a magnet. And then we go with this fragment after several steps of purification and amplification to the sequencing. The sequencing process, we first attach those fragments to a plate, and then we amplify the fragments. We generate clusters. We generate clusters because one molecule is not sufficient for seeing it, and we need a lot of molecules that are together in order to be able to see them with our machine. And then we start the sequencing. We anneal the primers, and then we add one nucleotide, we make a photograph. We add another nucleotide, second photograph, so we generate the base colors. And you see this is one photograph in the top of the slide, and here in this place we have an adenine, in this place in blue there is a thymidine, so we can see in this particular region this is the sequence we have read. And now we go for the alignment. This is the reference sequence on the top, and you see that there are no changes here, and here at this point there is a difference in the sequence. The number of times we are reading every base is what we call the coverage. And it's very important because, for example, you see there is one place that there is only a C. One C in maybe 30 or 40 reads. This is a mistake. This is an error, and it's not a real call. When you find several times, maybe 100% or 50% of the times, this is a real variation versus the reference sequence. Now this is the comparison of the technology. Here on the left we have our Sanger sequencing machines, and here our next generation sequencing machine. This is a high-seq, and if you compare, you need 65,000 Sanger sequencers to make the same job as one high-seq we have today. So we have an enormous capacity for sequencing. And we go from this situation, this is me in 2010, trying to fish the gene in the river, and now I go to the sea and I fish, I catch everything. The problem now is the interpretation. In our typical study, you may have 1,000, 3,000 to 12,000 variants that you have to classify and interpret. And many of these variants are variants frequent in the population that we call polymorphisms. Most of them not associated with the disease, but some variants that are relatively rare in the population may be associated with this expression. In the other side, variants that are very rare in the population we call mutations, may be disease-causing, but there are many mutations that are not disease-causing. And here, so there is an overlapping that makes it difficult sometimes to know what is pathogenic and what is non-pathogenic. So now, the interpretation, we need to be able to find the lobsters. We were not looking for them, but they are in the net. But we don't have to eat the rubbish. And this is the most difficult question today. For that, Health in Code, collaborating with Admera, we provide the best in the world interpretation tools because of our expertise and our multidisciplinary dedication to collecting all the data on diseases, patients, families, genes, mutations, papers. We have more than 30,000 papers in our database that have to be transformed for each individual, giving the information not only about the pathogenicity, but also about what are the consequences of the mutation or mutations that we can find. Risk stratification is our goal. It's important for the patient and the family to give a correct and timely diagnosis. And genetics gives not only diagnostic, whether this is pathogenic or this patient has or not has the disease, but what is the prognosis. And now I want to show you an example. I will skip this slide. This is a patient from our clinic. Sorry, it's not moving. This is a woman with previous diagnosis of hypertrophic cardiomyopathy. She's got severe biventricular systolic dysfunction and some thickening in the septum, some thinning in the posterolateral wall. And here you see this, she had to be transplanted when she was 40, 42, and had fibrosis, thickening, and some invaginations here. Her brother had a previous diagnosis of dilated cardiomyopathy, and this is the brother. If you see here, there is a biventricular systolic dysfunction, age 40, like the girl, but for me, the diagnosis clinically was left ventricular non-compaction because of this trabeculation. So it's a kind of non-compaction with severe systolic biventricular systolic dysfunction. Both of them have the same mutation in beta-myosin. This man had a girl, a daughter, 9 years old, and here you will see the daughter. Next slide. Okay. Sorry, I want to move it. This is the daughter, 9 years old, and she is also carrier of the mutation. The first question is whether she is clinically affected. She is completely asymptomatic, but for me there is a mild systolic anterior motion of the mitral valve, which is not diagnostic. Kind of shape of the ventricle, maybe something that hint of the potential development of hypertrophic cardiomyopathy, but this is really not diagnostic. But here you see the hypertrabeculation in the posterior wall, like the father. So for me, it's very suspicious that she's going to develop some kind of disease. And she's carrier of this mutation, glycine 716 arginine in the beta-myosin heavy chains. The question then is, what is the prognosis? Not only whether she's affected or not. If we make the survival curve of the family, you have nothing happens until they are 40 and then they are transplanted. We've got another patient with this mutation that has been transplanted in our hospital, which is a big transplant center. So what is the prognosis of the girl? If you go to the literature, in ClinVar, which is the database most companies are using now, some people classify as pathogenic and one group is classifying this variant as uncertain significant. There are 19 individuals and 10 families, and there is no information about the prognosis. In our database, we consider this pathogenic. There are almost 40 individuals, 19 families, and we have information about phenotypes, clinical details, and outcome, and there is prognostic information. And now I want to show you how we make this evaluation. Here I hope you can see this is our database, HIT mutations, and here we've got genes, mutations, we have articles, patients, families, and phenotypes and reports. So here, this is a system that will manage all the information. So for example, for this mutation, I will make a search. It is in the beta-myosin heavy chain gene, MY87. We have 17,000 genes in the database. The mutation name is glycine 716 arginine. So we search for this mutation in the system and we will find this information. Here is the mutation with different names, the pathogenicity, and here are the phenotypes. Here you see we have 42 carriers in 23 different families, and we have the phenotypes. And if we can go to the details here, related articles. For this mutation, in the literature, there are 50 papers, and we also have information of some families of our research, including this one. So if you want to read all these papers, it will take maybe a couple of months because you can read maybe one paper per day, but we read all the papers for you. And you take one paper, for example this one, and we can download the paper and read the document, and our inspectors and supervisors will introduce the data. Here, for example, this paper talks about several mutations, including the glycine 716 arginine mutation, which is our interest now. So, for example, in this paper, the inspectors who made the inspection of the paper, reading the paper, have introduced 4 different families. And you see this first family, this icon means that this family has been also in another paper, and the second family is in a different paper also. Sometimes, you know, the people publish the same families in different papers. Now, if you open here, you can see these are all the relatives that are quoted in the paper. And here, for each one of the individuals, we try to introduce all the available information. For example, demographic variables. This first patient is a male, Oriental from Korea, 37 years of clinical study, 23 at disease diagnosis, and 46 at the last follow-up. We have the genotype, his carrier of this mutation, and the phenotype. This patient had a diagnosis of dilated cardiomyopathy hypertrophic. This is the woman, or no, this is a male with both diagnoses. Environmental factors, alcohol, he was a heavy alcoholic. The echocardiography, he's got hypertrophy, 23 millimeters asymmetrical septal hypertrophy, no unknown wall thinning, left ventricular dilatation, 71 millimeters, systolic expansion, ejection fraction 16%, and there was atrial dilatation with 58 millimeters left atrial. We go to the symptoms, functional class 2, chest pain, not specified, and there is no syncope. The ECG, abnormal ECG with high voltage, and there is an abnormal Q wave, an atrial fibrillation flutter, and the events, and there's a heart failure death, age 46. Very remarkably, in this patient is similar to our patients in our family that I have just shown you. So with all this information, not only for the patient, but from all the relatives that are in this family, is collected and put together with the information of the other 52 papers talking about this mutation. So now we can go, I will go back to the mutation and see that after that, we, in our reports, not only say that the mutation is pathogenic, or very likely pathogenic, and in this case I would say definitely pathogenic. I will go back here, but we can even do this, create survival curves for the mutation. And I will show you this, which has not been seen by other companies. In this case, we can go for this mutation. I can filter for this mutation in the database. I click filter in, and here you see we have pathogenic and known pathogenicity and non-pathogenic variants. Only, in this case, it's only one mutation, 42 patients and 23 families. And we have the variance to the survival analysis. And here you see the Kaplan-Meier survival curve for this mutation. I will take out this filter. And here what you see is that putting together the data, we have 37 individuals providing information about survival. And after age 10, you have events in very young individuals that I can tell you these are sudden deaths, and this 30% sudden death before age 30. And especially it's an important proportion of sudden death between age 10 and 15, when you know that in hypertrophic cardiomyopathy many people think it's too early to make an evaluation. And all these are heart transplants or heart failure deaths. Only one individual with this mutation survived at age 55. So with this, we have additional information for this family. This girl, 9 years old, I have to be very aware of the risk of sudden death in the early phases of the disease. More interesting, sometimes this information is only about one mutation, and we don't know whether this mutation is really definitely important or not for all the family. So I can evaluate further details on the mutation, evaluating not only this particular mutation. I will clean the filter, but now I will go in to see other non-synonymous mutations that are affecting the same region. So this, what you see here, are the domains, functional domains of the protein that are affected by this mutation. And this is putting together information that is in the literature and is available there. So here you see this mutation is affecting the myosin motor domain of the beta-myosin. You know, beta-myosin is the main protein that generates the contraction in the muscle. Affects the head of the protein, but this is from amino acid 1 to 837. But also affects, for example, this domain, which is, I open it, this is the converter domain, which is a region important in fourth generation. And there are different definitions in the protein databases. So, for example, I can select this region and make the filtering of non-synonymous mutations that change the amino acid in this particular region. And here you see we've got 45 variants in 400 patients of 200 families. There are also rare variants that I will not include, and there is variants of unknown pathogenicity and frequency. And there are also sometimes, and not here, there are not variants that are non-pathogenic. So now we have all these pathogenic variants, and we can do the survival analysis of variants that are similar in the similar region, and you see here the survival is a bit better, but it's also quite a bad survival when you are 50 years old with these mutations, and here we are talking about almost 300 patients, your survival is 50%. Now I can even go to more detail. Which is interesting here is instead of looking for this region of the converter, I can see a more concrete region that is affected by this mutation. This helix between 715 and 721 amino acids, if I click here, you will see that we now only have 10 mutations, and there is one non-pathogenic, which is a synonymous variant not changing the amino acid, so this is not interesting. I will select here the protein level, sorry, non-synonymous. So we have 10 variants. We add these variants to the survival analysis, which are very close to this mutation, and you see with now 147 individuals, it is confirming the high risk of this particular small region of the protein. So here we are giving prognostic information. As an example, I can show you, for example, the survival of all the patients with non-synonymous variant in the beta-myosin heavy chain gene. Let's go for that. I clean this filter, and then I say protein level, non-synonymous variants, and now all the gene. And now the system takes a bit more time because they have to evaluate thousands of genetic variants. And here is the difference between other people and our system. We can differentiate, even within the gene, prognostic information. While the system is loading here, you see, now we've got more polymorphisms. We are not interested in polymorphisms. I will click them out. Also, variants that are non-pathogenic in that region, and take out also variants of unknown significance, and I only leave those mutations that we consider pathogenic. Almost 3,000 patients, 1,600 families, and here you have all the mutations. So all this information has been introduced with detailed data of many of the individuals, and now we can generate the survival analysis for these beta-myosin heavy chain mutations. And this is what we try to do whenever there is information. And it's very important because if you consider beta-myosin are one of the most frequent genes in hypertrophic cardiomyopathy, and not all the mutations are the same. So we go here, and you see, if you compare the survival curve in the red, which is all the beta-myosin, with the survival curve of my patient, you see that we are dealing with a patient which is very special in this population. And I don't think we have much time to go into more details on how we deal with this, but just to show you, we go to when we have our own patients, we can manage all this information together to generate the reports. So here we are going out of this particular. Here we have, I didn't show you, the p-values comparing the different survival curves. So all this information is provided whenever possible in your reports. So I will go back to my presentation. So whenever I just, in this particular mutation, then affecting the converter region and this particular helix, now we know that for this girl, and this is the girl 3 years later, this is a typical hypertrophic obstructive cardiomyopathy. She's 13 years old, 67 millimeters of mercury. And with our knowledge about this particular mutation, now we decided, when she was 15 and we found only one run of non-systemic tubular tachycardia, to put a defibrillator to prevent sudden death. Now, several years later, she's 20 years old and she's developing systolic dysfunction. Unfortunately, she's not still alive. So we don't have today much time, but if you are interested, we can prepare other webinar about other conditions. In all these conditions, it works the same. You put together the data either in long QT or in Marfan. For example, in Marfan, where you know that there are different regions in the protein, there are different types of mutations with different prognosis and evolution, and this is important for the patients and the families. So my conclusions is that genetics are necessary for a correct diagnosis and clinical evaluation of patients with inherited cardiovascular diseases and arrhythmic syndromes, and the guidelines also support the use of genetic testing. NGS offers the best quality and cost efficiency, but a correct clinical interpretation is essential for the correct application of next-generation sequencing in clinical practice. So I want to show you our differentiator are the reports Health Encodes providing with Athmera in our CLIA-approved studies. We provide in this first page all the summary that you need if you want a very simple diagnosis, but you have also the details and a clinical interpretation giving the data on the clinical bioinformatics and conclusions and the tables with the data of the patients, and whenever possible the survival curves giving information about the prognosis, not only in the short but also in the long term, that you have to put together with the clinical data. Thanks for your attention. For any questions, I will be available at my webpage and in Athmera Health. You can also contact us. And I invite you to try our service, because I tell you we will provide the best interpretation, which is key for your patients. Thank you very much.